Hello everyone, we will start with another session of regeneration biology, and this section is tissue regeneration in mammalian context. And we know that mammals have lot of advantages when it comes to survival in the planet, but when it comes to regeneration, mammals often we will find them as little debilitated compared to non-vertebrates. So mammals in general are excellent in a lot of their protection against pathogens. They have a supreme immunity, etc. But when it comes to the regeneration, it often lags behind in a lot of complex tissues. Simpler tissues, it do regenerate like skin or nail, or if there is a bone fracture, it can heal, etc. But when it comes to complex tissues, say brain, spinal cord, heart, kidney, it struggles. So let us see what are the possible causes behind it. So the regenerative phenomenon is widespread, but the regenerative capacity varies greatly among animals. Invertebrates, that means the ones which do not have the backbone. Vertebrates groups are mainly 5, starting from fishes, frogs, reptiles, birds and mammals. They are the main 5 groups of vertebrates. So phylogenetically primitive vertebrates such as salamanders, zebrafish, mainly fish and amphibians, they possess a higher regenerative capacity than mammals. Even in the same individual, different tissues or organs, they have distinct regenerative capacity. For example, in our body, the liver can regenerate excellent, but the heart or spinal cord lags behind. In addition, the younger animal is usually easier to regenerate than the older one. Decades of research are beginning to explain why regenerative capacity differs markedly among different cellular and molecular components, and can it also have got some evolutionary implication. Exploring the reasons for differences in the regenerative capacity has resulted in finding the causes such as the properties of stem cells, which tissue you are talking about, or the ability to dedifferentiate or transdifferentiate for a given tissue cell, or expression of specific regeneration associated genes, lot of epigenetic regulators, and how the immune system contributes to the regeneration. So the comprehensive analysis of these perspectives would allow us new insights into how to promote regeneration in mammals. So this picture you can see, it's a schematic of regeneration in various animals. Say planaria, you can take a planaria and cut into pieces, as many pieces as you want. Each piece will give rise to, say this is cut into 1, 2, 3 pieces, it gave rise to 3 planaria. Same way Hydra, another, it has got only 2 germ layers. It's another primitive invertebrate. Planaria also is an invertebrate. It can cut into any number of pieces and it regenerates. And now coming to vertebrates, lizard's tail or limb, you would have seen, if you cut the limb, it grows back. Or even tail, you may have seen wall lizard shedding its tail and growing back. And when it comes to xenopus, normally if you cut a tail or limb or any other body part, it normally regenerates back. And in zebrafish, it has been extensively studied for the heart regeneration and retina regeneration and spinal cord regeneration, brain regeneration, etc. In this picture, the heart regeneration is shown. And in the case of mice, the only organ, complex organ that can regenerate, mice is a representative of mammals, liver is the only organ that is capable of regeneration. A few tissue-specific stem cells are preferentially preserved in certain high turnover tissues of adult mammals. What does it mean? Say your intestinal epithelial cells, skin epithelial cells, they are prone for damage. They are blessed with having stem cells underneath. So if there is a old skin is shed, the new skin comes underneath. So for example, human skin, blood system, bone marrow stem cells constantly keep producing your blood cells. They have the capacity to regenerate which is largely because of the reserved epidermal stem cells in the case of skin, and also hematopoietic stem cells are present inside your bone marrow. Unfortunately, most adult mammalian tissues have few or no resident stem cells to support them. This is likely one of the major limiting factors to regeneration. We have explanation for why there is no regeneration, but are these explanations sufficient enough? It is almost like telling, why are you climbing Himalaya? The answer is yes, it is there. That is why I am climbing. That is not the answer. Same way, stem cells are not there. That is not the answer, because when it comes to lower vertebrates, they are regenerating in spite of not having dedicated stem cells underneath their respective tissue. If, however, the small number of resident stem cells in the adult mammalian tissues could be stimulated and recruited, it is possible to promote regeneration. So this is the conjecture one has to test. For instance, the mouse can achieve digit-to-tip regeneration by stimulating few distinct lineage restricted progenitor cells, and it can form a blastema-like structure, so that even if your finger is cut, if the nail bed is preserved, then there is a good possibility that the fingertip can grow, but it may not be as long as your digit. But if the nail bed is not restored, then chances of fingertip appearing or growing back is limited. So let us understand what is dedifferentiation potential. It's a new terminology what you are learning in simplistic sense. I'm sure all of you who are taking this class would have completed your +2, and or maybe you have completed your bachelor's or even master's. But let us take a bracketed group. All of who are taking this class would have completed your +2. Means you are all 12th pass. Now, if you are asked to go and sit in your kindergarten, say UKG, LKG or class 1 or class 2, what do you understand by saying that? Means you are erased with all your knowledge or whatever learnings happened this many years, and you have been asked to learn right from the kindergarten, say alphabets, number, counting, writing, etc. Something like that is in a simplistic form dedifferentiation, that is going backward in time. Mammals fail to regenerate bones after the amputation of their bones, although internal bone defects can be healed below a critical size. So, if the bone has a severe damage, it cannot heal, but if there is a fracture, there is a crack, and you put these bone parts together, it grows. In mammals, what happened? The de novo osteoblasts deriving from the mesenchymal stem cells contribute to the bone healing process without osteoblast dedifferentiation. This is what's been observed in the case of mammals. The scarcity of dedifferentiation in mammalian osteoblast may be the underlying reason why the bone cannot regenerate. Say you removed a bone, new bone cannot form, or the damage to the bone is very severe, it cannot be fixed. So there can be a limited amount of damage to the bone, it can heal, rather it can fuse. So retina regeneration in zebrafish is a good example, and even chick, neonatal chick, soon after birth, the chick also can, soon after hatching, chick also can regenerate its retina. And the mammals, it often fails in doing that, and depends on the dedifferentiation of the Mueller glia. Zebrafish or chick or any animal that is capable of regenerating the retina depends on the dedifferentiation of the Mueller glia, although the mammalian retina has a much lower regenerative capacity than that of zebrafish and chick. What does it mean? It simply means that they do not know the exact mechanism, exact processes through which it can reprogram or dedifferentiate. This is probably ascribed to the lower dedifferentiation potentials in the Mueller glia in mammals than in zebrafish and chicks. Having said that, you should also understand less capability means less instructions are gone onto that Mueller glia. Mueller glia is a cell and it is ready to express any gene, but it is not been given the proper instruction. You should read in that way. Not that the Mueller glia cannot reprogram, it do not reprogram. So that is what we should understand. Although most mammal tissue cells lose dedifferentiation potential, several cells retain this ability. For instance, Schwann cells undergo dedifferentiation for the peripheral nerve regeneration. Say you got a burn wound and a new skin is being formed, and for few days you will not have the sensation, but after a few months you will retain your ability to the touch. If someone touch, you can sense. And same way, people who get leprosy, they lose their sensation on the skin because the Mycobacterium leprae gets rid of it damages your peripheral nervous system. But if it is healed, it can bounce back thanks to Schwann cells, which are the glial cell population of the peripheral nervous system, and they are called Schwann cells. Although mammalian cells cannot take natural dedifferentiation after injury, dedifferentiation can be induced in vitro. Mouse myotubes are induced to dedifferentiate and proliferate after treatment with extracts from the regenerating limbs of newts after ectopic expression of one transcription factor that's called MSH homeobox, also known as MSX1. So it can be induced. This indicate, like I told you, it's not that it cannot, but it do not dedifferentiate naturally. This indicate the mammalian cells, like myotubes, have the potential to dedifferentiate, although the potential need to be stimulated or it need to be induced. Why do the cells of the primitive vertebrates such as fishes and frogs undergo dedifferentiation more easily than what you see in the case of mammals? That is the question. Although the specific mechanisms are not clearly understood, cell cycle regulators play a major role. They are found to play an essential role controlling the dedifferentiation process. Terminally differentiated, that means from stem cell up to final differentiated cell. Finally differentiated cell, they are called terminally differentiated. Newt myotubes, that is, newt means basically frog, amphibian species. Myotubes can dedifferentiate after injury because of tumor suppressor retinoblastoma RB. RB proteins are phosphorylated and they are very important to regulate your cell cycle, thereby allowing cells to re-enter the cell cycle. Means cells which are arrested in cell cycle, that means normally it is called G0 arrest, they can enter back and now they will become G1. However, mammalian myotubes do not phosphorylate RB proteins after an injury and fail to re-enter the cell cycle, and one of the causes why it is not able to dedifferentiate. This suggests that RB phosphorylation may be a crucial barrier for mammalian muscle dedifferentiation. In support of this, do we have any evidence? Transient inactivation of RB and the alternative reading frame, ARF tumor suppressor, another protein, forces the mammalian myotubes to re-enter the cell cycle and lose the differentiation properties. Means myotubes can be pushed into dedifferentiated phase if you inactivate the RB protein. If it is not spontaneously happening, forcefully you inactivate, then you can push them into cell cycle. In addition, RB and another RB family member P130, that is molecular weight is 130 kd, can block cell cycle genes and maintain the post-mitotic state of the mammalian adult cardiomyocytes. Knockdown of RB and P30 together leads to cell cycle re-entry in the case of adult cardiomyocytes of the mammals. As a cell cycle inhibitor, P53, one of the well-studied famous tumor suppressor protein is P53, also hinders the differentiation. That means during salamander limb regeneration, an early downregulation of P53 is a prerequisite, means essential for mesenchymal cell dedifferentiation and blastema formation. In our own labs research, we have seen another tumor suppressor called P10, phosphatase and tensine homologue, spontaneously gets downregulated during retina regeneration. If you prevent that, then you don't get retina regeneration. So what does it mean? That tumor suppressors also contribute in making the cell dedifferentiate. Because cell cycle inhibitors block dedifferentiation in mammalian cells, targeted modification of these inhibitors will likely promote dedifferentiation and regeneration. In addition to these cell cycle regulators, several epigenetic factors also contribute. They control the differentiation and maintain the differentiated status. They are also important. If any tweaking happens to these epigenetic factors, they can push them into dedifferentiation. So sometimes a tissue need to be maintained in differentiated status. So epigenetic factors do contribute. It can be histone modifications such as histone methylation, histone acetylation, etc. Targeting the epigenetic regulators has been applied to facilitate dedifferentiation. For example, the forced expression of transcription factors or the treatment with small molecules that can change the epigenetic regulators such as DNA methylation or histone modification result in the complete dedifferentiation of the somatic cells into pluripotent cells. So, these are some of the interesting observations that is being shown by artificially tweaking the scenario for a mammalian cell. Accordingly, the dedifferentiation potential may be enhanced artificially by targeting cell cycle regulators or epigenetic regulators. So then comes the transdifferentiation potential. What is transdifferentiation? Dedifferentiation we have seen, that is going back in time. That means a intermediate student going to kindergarten. Now you are not going all the way to kindergarten. You are, say you are a bachelor student. You have studied biology for your BSc. Now you are going up to +2, and instead of studying BSc biology, you are now going into medicine, or you are going into LLB, or you are going into engineering. That means it appears like a person who was studying BSc biology, it now became a lawyer, became an engineer. That is called a transdifferentiation. You have to go back in time, but no need to go all the way back in time. So for example, newts and frogs can completely regenerate their lenses via cellular transdifferentiation. In newts, once the lens is removed, pigmented epithelial cells from the dorsal iris, dorsal and ventral, dorsal means top, ventral means down. So in your back and front, if you see in that angle, so dorsal ventral concept you can see in the book. So basically, if I want to express your dorsal and ventral, your back is your dorsal, your chest is your ventral in a simplistic term. So the dorsal iris transdifferentiate into lens cells and regrow the entire lens if you remove the lens in your eye. That is the one or part which is removed during cataract. If your lens become opaque, you can't see, and in cataract surgery you remove the opaque lens. Okay, that is lens. Similarly, the frog lens can regenerate through the transdifferentiation of the corneal epithelium into lens cells during the larval stage. In contrast, the mammalian lens only has the ability to achieve incomplete regeneration from lenses on epithelial cells without the transdifferentiation of other cells. Mammals lag behind in here too. Therefore, the loss of natural transdifferentiation in mammals impedes complete lens regeneration. However, mammalian cells retain the transdifferentiation potential, which has to be followed up further and see whether can we make through an exogenous stimuli, can we make a lens. Recent reprogramming strategies have extensively done to confirm the latent transdifferentiation in mammals can happen, do exist. Somatic cells such as fibroblasts can be induced into another lineage. For example, neurons, cardiomyocytes, hepatocytes, anything you want, you can convert by several reprogramming approaches. And this include the production of a lineage factor based reprogramming, and we also call them as iPSC, induced pluripotent stem cells. And this is basically you are directing a fibroblast or a normal cell into stem cell. These reprogramming approaches, especially small molecule mediated reprogramming, will offer very meaningful opportunities that allow to deliberate transdifferentiation of one cell type into another, either in vitro or in vivo. So this is a picture that shows how the transdifferentiation is done or the reprogramming is done. You take a somatic cell and you can convert them back due to various signaling event using either small molecule or using transcription factor. Ideally, you can make iPSC based reprogramming where you make them the somatic cell into iPSC and it can give rise to any cell through differentiation, or lineage factor based reprogramming where the somatic cell you are directly converting them into a cardiomyocyte by expressing cardiomyocyte specific factors in them, or neuron or cardiomyocytes, anything. So this is very much possible in vitro, but in vivo the research is still going on. So the regenerative species depend on the ability of adult stem cells in their body or have a huge potential to undergo dedifferentiation and transdifferentiation in the adult cells. So this is the hallmark of regeneration, and mammals often lag behind. However, a few mechanisms are lacking in most adult mammals, limiting their regenerative capacity, such as activation of stem or progenitor cell. We know progenitor cells are needed, but are they able to make it? The answer is no. The reversion of differentiated cells into their progenitor, that is called a dedifferentiation, and it do not happen effectively. The conversion of one tissue into another, that is transdifferentiation. So these are the limiting factors when it comes to the mammalian regeneration. The selected expression or silencing of regeneration associated genes also affects the regeneration, and the genes might be possessed or expressed exclusively in a regenerative species but not in a non-regenerative species such as mammal. In recent years, accumulating evidences have strongly suggested that the epigenetic regulators that exert enormous influence on the so-called regeneration associated gene expression, and they can modulate the epigenetic landscape and they can influence the regeneration. And let us see what are the regeneration aiding genes. Almost all animals can heal wounds, but only some can regenerate. We know that. One hypothesis is that certain regeneration specific genes are expressed exclusively in regenerating species. Let us see whether it is true or not. One salamander specific gene, say PROD1, PROD1, which encodes the glycophosphatidyl inositol anchored protein. It is found to support this hypothesis. What does it say? PROD1 is expressed in the blastema and is essential for patterning of and growth during the salamander limb regeneration. More importantly, no ortholog of PROD1 has been found in xenopus, zebrafish or mammals. So what does it tell you? This PROD1 is a unique feature found in salamander which can regenerate a damaged organ. Therefore, the regeneration gene is specifically possessed by salamander. One could argue, but that is not always the case. But this is one example of a gene. Another possibility is that non-regenerative and regenerative species both carry certain regeneration genes, but these genes promote regeneration only in a regenerative species. The same gene is not promoting regeneration in a non-regenerative species. So this is another possibility. Let us see whether is there any evidence. This possibility is verified by specific expression of growth factor FGF20A in zebrafish. FGF20A is expressed early after fin amputation and initiates fin regeneration. By contrast, the ortholog of the same gene FGF20A in mammals is not associated with regeneration. It is not turned on. It is not contributing to regeneration. It is worth mentioning that selective expression of regeneration genes affects the regenerative capacity of different tissue and even in the same animal. So we cannot generalize that this gene must help in regeneration or this gene is essential for regeneration. Nutelents, we already discussed about the nutelents, can regenerate from pigment epithelial cells of the dorsal iris but not from the ventral iris. Both are iris, one is dorsal, one is ventral. What is so much difference between them? One study has shown that this situation is due to a deficiency of one lens specific regeneration gene, that is sine oculus related homeobox, also known as 6-3 in the ventral iris. Dorsal iris have 6-3, ventral iris don't have if you remove the lens. 6-3 is required for the lens development during embryogenesis but is only expressed in the dorsal iris after the removal of the nutelents. Then what people did? If you express 6-3 in the ventral iris by transfection, it generates the lens. You don't need dorsal iris. If you get rid of dorsal iris, ventral iris can give rise to all it wanted was the 6-3 gene. Altogether, certain species and tissue, like you said about the dorsal and ventral, both are iris, but dorsal iris versus ventral iris, have high regeneration, have special regeneration specific gene in a given species or a given tissue after injury. So now let us see immune responses. The more phylogenetically primitive urodale amphibians, such as salamanders, they appear to have a weaker cellular and humoral immune responses regarding the specificity, speed of onset and memory compared to that of a anurine amphibian. That is normally frogs which is living in the land compared to a their siblings or their related members who are living in water. In response to the larval stage, it is relatively ancestral and much less well developed than that of the post-metamorphic stage, which has highly evolved. As the organism evolves, they have a better matured immune response than the larval stage. In mammals, the transition from fetal scarless wound healing to adult typical scarring is accompanied by a gradual increase in inflammation and also this is contributed by the immune cells and also by various pro-inflammatory cytokines. These studies indicate that animal regeneration or animals' ability to regenerate loss relates to immune systems development. More advanced immune system is not allowing you to have a proper regenerative response. The immune response does not always hinder regeneration. One should not think that, oh, if immune system is powerful, you can't get regeneration. But there is a catch behind it. Successful regeneration, in effect, demands proper immune response. You should know when to give acceleration, when to give the break. This is what is understood. For example, immune responses are indispensable to salamander limb regeneration and neonatal mouse heart regeneration. If you interfere with the immune system, both cannot regenerate at all. That means immune system is needed. Common functions of the immune responses in regeneration include scavenging the cellular debris. Whenever there is an injury, there is a debris, and activating the progenitor cells and promoting angiogenesis. That means the blood vasculature formation. Many immune cells, such as cytokines and components of, are engaged in these processes. Among them, the macrophage response is very classical. They play an important role in the regeneration. After amputation of the axolotl limbs, macrophages are recruited early into the regenerating blastema. In contrast, the systemic depletion of the macrophages leads to failure of the full limb regeneration and it leads to extensive fibrosis, as you can see in the case of a human limb or a mouse limb damage. So the tremendous strides have been made in delineating the regeneration process and the cellular and molecular mechanisms of regeneration in animal models. After comparing many aspects of animal regeneration, it could be suggested that the several possible reasons could exist to explain why regenerative capacity differs among animals. Those reasons may account for the low regenerative capacity as observed in the case of mammals, and they can provide novel avenue for promoting the regeneration in mammals. Because the adult mammals, they have insufficient stem cell, one could always tell why it is not able to regenerate effectively. The induction of dedifferentiation and transdifferentiation is crucial for obtaining cellular sources for regeneration. You want to make a building, you need to have brick, and for regeneration, the bricks are nothing but cells. In high stability of adult mammalian cells, they can prevent them from changing their cell states, which considerably restrains their dedifferentiation and transdifferentiation potentials. So many research studies have shown that in a neonatal pup or a postnatal chicken, the regenerative capacity is tremendous. Whereas the same animal, same species, once it differentiated, the immune system advances. Although immune system is necessary, the advanced immune system make the environment rather more pro-inflammatory, and this can interfere with the ability to regenerate effectively. So what we should understand from here, as organisms grow older, the macrophages we already discussed. There are two types of macrophages, M1 and M2. M1 is always pro-inflammatory, whereas M2 is always anti-inflammatory. The levels of M2 macrophages depletes as the organism grows older and older. And M2 macrophages are very much low in the case of diabetes patients, which could explain why they are having too much of ulcers and difficulty in healing. Even a simple wound, it is not a regeneration. So we'll study more about regeneration in the next class. Thank you.